Good morning. Good morning, everyone. Good morning. Good to see everyone. We're still a minute or two before 10, so we'll give people a chance to come in. If you would, have your video camera on. It should be on for you already. And mute your microphone. I find that our meetings will stream best if we stay muted, unless we're speaking, unless you want to ask a question, or if I ask you a question. So it looks like a lot of people are here, which is great. What I'd like you to do first is go ahead and type your nickname or your first name into the chat for me. That's the way I take attendance. Thank you. Very good. That's great. Thank you. That'll be my record that you attended today in our Zoom session. So thank you very much. So for those of you who haven't met me before, my name is Dr. Piscopo, and I'm going to be your instructor this semester for microbiology, specifically microbiology for healthcare professionals. So a lot of you have told me that you're interested in nursing, that you're planning on earning some kind of nursing degree. You're hoping to go into clinical practice with nursing, which is great. Some others of you told me that you are interested in other public health fields, that you're hoping to get involved in public health in some other way. So I will do my best this semester to sort of gear our class towards what everyone's interested in. And we'll try to bring in examples and things that help us learn these important concepts in microbiology. Most of you have not taken a microbiology class before, which is absolutely fine. This is what we call a survey course in microbiology, which means we're going to learn a little bit of everything in this course. So I think you're going to find it's an interesting course, and it's very, very practical for people who are interested in public health and who are interested in healthcare. It's one of those topics that touches on many other different topics in science. And it's very, very practical. We use it every single day in diagnostic laboratories across the country. And of course, right now, in the middle of a pandemic, we're using a lot of microbiology every single day as we talk about the SARS-CoV-2 virus. So yeah, we'll bring the pandemic in as well whenever we can, and use it as an example as we learn this subject. So if you haven't already, go ahead and type your first name or your nickname into chat for me. And that'll be my record that you attended this morning to Zoom. And yes, we are remote this week as we come back from our winter break, but that's actually a really good strategy from a public health perspective, because it'll give us a chance to take a surveillance approach to testing and see who might have gotten exposed and developed COVID over the break. You probably already know that we've adjusted, the college has adjusted its COVID protocol a little bit. This semester, we're going to be taking into account whether or not you are vaccinated in terms of what you will need to do if you test positive. So lots of things happening this semester on campus to try to keep everybody safe. So we'll be remote this week, today and Thursday, and then hopefully we'll be back in the classroom next week. And what we're going to be doing this morning, we're going to have our first lecture topic this morning, which is really just an introduction. It's an introduction to microbiology, an introduction to the microbial world. But we'll spend a few minutes first just talking about the course. Now, hopefully you've had a chance to go into Canvas and to go through the start module, the start here module in the course. This course is completely organized within that modules section in Canvas. I think a lot of courses now are using modules as the sort of the organizational home base for courses. So we're going to do that as well. And you'll see that the course is organized week by week by week in Canvas. And at the beginning of each module, there's going to be a to-do list for you. And I hope that's helpful to you. At the beginning of each week, you'll be able to click on the to-do list and get a nice summary of everything that's going to happen that week in the course and everything that you're expected to do that week in terms of, you know, lecture homework or a laboratory assignment or whatever. So it's going to be really important that you get in the habit of clicking on that to-do list each week before we get started into our week of work. So just to introduce myself, for those of you who don't know me already, I am a veterinarian. I'm a licensed veterinarian. I don't practice anymore. I'm retired from clinical practice, but I did practice in what's called a mixed animal practice. That's the kind of veterinary practice that sees all different kinds of animals. We'll take any kind of animal that walks through the door. I grew up in Massachusetts, and I did my undergraduate work at the University of Vermont. I studied animal science. I got my degree in animal science. And then I went to veterinary school in North Carolina. I went to North Carolina State University to go to veterinary school. And when I graduated, I stayed in North Carolina for about a decade. And again, I worked in a large veterinary practice. I loved it. It was everything I had hoped it would be. My particular area that I did most was horses. I did equine medicine primarily. So I had intended that I would practice forever, but things changed for me, as they often do for us. When I started having kids, I had an opportunity to go back to school. I was offered what's called a fellowship to go back to school and get a PhD. At the time, I was working about 100 hours a week, and that included a lot of overnight emergency duty. And I was having young children, so I took the opportunity to go back to school. I earned a PhD in physiology, human physiology, and then I had intended to go back into practice, but I was injured. I injured my spinal cord pretty badly, and I had to retire from practice, which was a great sadness to me. But luckily, with my excellent education, there were other things I could do, other things I could try. And one of the things I really wanted to do was teach. That brought me back up to New England, back to New Hampshire. And I got my first job at Keene State College. I'm at what's called an adjunct professor. So I do not work full-time at Keene State. I'm part-time at Keene State. I'm part-time at other colleges in New Hampshire. That's what brought me here. I teach a little bit of everything. I teach microbiology, I teach biology, I teach evolution, genetics, cell biology. I teach a course in reproductive biology. I teach a little of everything, and I really enjoy teaching. It's really a passion of mine. So now you know a little bit about my background, and it'll make sense when I bring in animal or veterinary examples of things. That's where my wheelhouse is. So, all right. So I can see that most of you have gone through the Start Here module in Canvas and have done that student survey that's in that Start Here list of instructions. That's great. Thank you very much for getting that into me. If you haven't done that already, please try to do that and get that to me this week. Okay, that survey is just going to help me get to know you as a class, so I know how to gear the course a little bit and make it relevant to you as much as possible. When you go into Canvas, underneath that Start Here module, you'll start into the weekly modules, including the module for this week. And our schedule this week is to have a lecture today over Zoom. And then on Thursday, we're going to do a laboratory exercise together over Zoom. We're going to talk about biosafety in the laboratory, very important topic. And we're going to talk about the care and use of microscopes, because we're going to be making heavy use of our light microscopes this semester in class. And like I said, hopefully next week we'll be back in our classroom on campus, sorry. I have Canvas on the brain. You do have a lecture homework to do this week. You will have a lecture homework most weeks. And when you go into Canvas, what you're going to notice is that it's set up like a quiz. I call it a homework, but it's set up the way a quiz is set up. And that just is how Canvas works. That's how Canvas works. So you'll have all the time you need to do the assignment, and you are free to use your notes when you do lecture homeworks. The purpose of the lecture homework is to make sure that you understand what we talked about in lecture. So you are allowed to use your notes. You're allowed to take as much time as you want. The one thing to remember about these homeworks is that you can only submit it one time. Okay, so you have to make sure that it's how you want it, and then you submit it, and then you're done. It gives you some automatic feedback, right? If you've done quizzes in Canvas before or exams in Canvas, it's going to tell you right away for multiple choice questions which ones you got right and which ones you got wrong. So there's no opportunity in this class to do a homework more than once to try to improve your grade. All right. So that means something. That means you want to make sure that your notes are in order and you're ready to do the homework. And when you sit down, you're going to want to finish it. However long it takes you, you're going to want to finish the assignment and then click that submit button and you'll be all set. You're also going to have assignments called lab homeworks. When we have laboratory exercises, there'll be some homework questions for you to think about and answer. And it's a very similar setup. You're allowed to use all your notes, all your data that you gathered in laboratory. You'll be able to take as much time as you want, but you'll need to submit it. You'll need to sit down, finish it, submit it, and you're only allowed to do it once. Now, the other ways you're assessed in this class, of course, there will be examinations, and you can see the schedule of our exams in our syllabus. The other thing that I do periodically is what's called a pre-laboratory quiz. I will let you know if we're going to have a pre-lab quiz. But all that is about a 5-minute quick quiz at the start of a laboratory, which tests whether or not you read the protocol for the day. You guys are all second year and above, so you are certainly capable of preparing for laboratory now. And the way I ask you to prepare is to read the handout that I provide for you on Canvas, so that when you come into lab, you're at least aware of what we're going to be doing that day, what the purpose of the lab exercise is, and what kind of techniques we're going to be learning, and so on. So we're not going to have a pre-lab quiz for every lab, but periodically I will do that, and I'll let you know when we're going to have one. So what I'm going to do next, I'm going to go ahead and I'm going to pull up Canvas for us to take a look at. I'm going to pull up our home page on Canvas. Bear with me for just a second. I get rid of these other things. So hopefully that's what you see right now on your screen. Hopefully you can see it says Microbiology for Health Professionals up at the top. This is our home page in Canvas. And I like to think about this as our home base, because there's some information on this page that you're going to want to refer to again and again as we go through this semester. For example, you can always find a copy of our syllabus on the home page. I keep it up to date. If I have to make any changes to anything in our schedule, I'll do that, and then I'll repost it right here. So you'll always have an up-to-date copy available. You've got some information about Zoom here, in case we have to go back onto Zoom outside of this week. And then there's a little bit of information now about our first week. Again, today we're going to have a lecture called Introduction to the Microbes. And then on Thursday this week, we're going to have a laboratory called Biosafety and Care and Use of the Microscope. One of the things that we're going to do in this class that you may not be familiar with, you may not have experience with, is we're going to make use of recorded lectures as well as live lectures in the classroom. And what I do is I record a lecture topic and I post it up on YouTube. I post it in a playlist on YouTube that is just for our course. And right now there is a playlist that is dedicated to PH240, Microbiology for Healthcare Professionals. And you'll find recorded lecture material in there. Now, of course, most of our lectures will be in the classroom, live in the classroom. But periodically, you'll be asked to watch a lecture from home, usually because we're going to spend class time doing something else. I'm going to pull my screen back up again, share my screen again. If you look right here on the screen, there's a couple of links. It says our YouTube channel, and it happens to be called Science on YouTube. And then more specifically, our playlist. If you're not particularly familiar with YouTube, people who make channels on YouTube can also set up playlists where they post particular videos. So our playlist is named for our course, Microbiology for Healthcare Professionals. And if I click on that link, I'm going to be brought into YouTube. And hopefully that's what you're seeing right now. So you can see there's a video loaded up right here, ready to go. This happens to be the one we're going to do today, the lecture we're going to do today, Introduction to the Microbes. You can see the name of the playlist right here. And then I can scroll down this list and I can see all kinds of lecture videos. All right. Again, most of these you're not going to watch in video form. Most of these we're going to do live in the classroom. But for example, next week, let me see if I can scroll up here. Next week, you're going to watch this video from home. You're going to watch History of Microbiology and Science of Microscopy from home. And you can see that there are 4 individual sort of chapters to this particular lecture. It's split into 4 videos. All right, so you'll want to watch video 1, then video 2, then 3, then 4. One of the nice things about watching a lecture from home, I think, is that you can stop and start it if you want to. While you're taking notes down, you can stop and start it. And you'll also have it available to you later on, so that while you're studying, or maybe making an outline for yourself, or making some study materials for yourself, you can always refer back to the lecture by pulling the video up. Sometimes having it recorded is actually gives you a little bit of a leg up, as opposed to only getting to hear it once in the classroom. So that's our playlist on YouTube, and that will be available all semester. Now, there may be times this semester, unfortunately, when you can't come to class because maybe you're ill, or perhaps you are in quarantine or isolation due to COVID. Maybe you've had a positive test and you're not feeling well. Maybe you've got some kind of a personal emergency, heaven forbid, that you have to tend to. If that's the case and you cannot come to a scheduled class, you're going to want to message me right away and let me know that you can't come and why. But the other thing that the playlist will allow is for you to view lectures that you have to miss because you have to miss class. So if you find yourself in isolation or in quarantine or out of class for any other reason, you'll have access to a recording of the lecture that we went through that day in class. And hopefully that'll be a help for you as well. Now, just a reminder, our COVID policies have changed a little bit this semester. So we still don't want you to come to class if you're sick. If you are sick, you should stay home. And that's whether you have COVID symptoms or any other symptoms. If you're sick, you need to message me and then stay home. And like I said, you'll have access to lecture material on our playlist, so you don't have to worry about missing lecture. If you miss a laboratory exercise, due to illness, you'll have to get the notes and the data for that laboratory from a classmate. When we get back together in class next week, we will exchange information with each other. We'll exchange email addresses and things like that, so that you can get up with somebody if you miss a lab, and you can get the information you need to complete the assignments for the lab. So the take-home message is, if you're sick, stay home. Now, if you test positive, but you are vaccinated, you are welcome to come to class if you have no symptoms. If you test positive and you are asymptomatic, and you're vaccinated, you are welcome to still come to class. Because of course, we now understand, after 2 years of this pandemic, that COVID is a very different disease in vaccinated people versus unvaccinated people. So vaccinated students who are feeling fine but test positive are allowed to come to class now, as long as they're masked. And of course, we're all going to be masked this semester. So that's good news. Hopefully we won't need to miss as many classes as we've had in the past. So I'm going to pull up our Canvas page again. We'll take a look at a couple other things. What I've got on the screen now is our syllabus. And what I want you to notice is that there's a lot of information repeated on the syllabus, including the links to go to that YouTube playlist and all kinds of information about what things we're going to be learning in class. The breakdown of what your grade is going to be based on is in the syllabus. The grading system that we use in terms of your course grade, on and on, all kinds of information. There is a couple of things I do want to draw your attention to. I'm going to scroll back up to the top and you'll see this attendance policy. Okay. It's important that you know that if you miss class, the class is not going to be broadcast over Zoom. All right. We're not going to be broadcasting live over Zoom this semester. Unless the campus pivots to remote instruction again. Okay. If you're unable to come to class, you need to message me. And by messaging me, I mean use the inbox tool in Canvas. That's an important thing to note down today. Instead of just sending me a straight email message, which you might be used to doing, I want you to do it through Canvas. Go into Canvas, click on in the menu where it says inbox, and then send me a message that way. Now, let me tell you why I want you to do that. When you send your message to me through Canvas, I get it separate from all my other emails. And as you can imagine, I sometimes get hundreds of emails every day. And so it's hard for me to catch student emails within all the rest of those emails. If you message me through Canvas, I will see your message because I get it separately from all my other emails. All right. So the very best way to get my attention, to get in touch with me, is through Canvas. Use that inbox tool for me. All right. So the attendance policy is important to note. The other thing that's important to note is that we're going to be using a new application in Canvas this semester called Lockdown Browser. When we take examinations, we're going to be in the classroom, but we're going to be doing them on our laptops. We're going to be taking the examinations in Canvas on laptops. And what's going to happen is you're going to have to click on the exam using an application called Lockdown Browser. What this does is it adds a layer of security to the exam. Like the name suggests, it prevents students from opening up any other windows on their laptop while they're taking the exam. Now, I don't expect that any of you are cheaters or that you're going to try to cheat on an exam. I don't expect that from you. But unfortunately, there are some courses and there are some students who will absolutely try to take advantage during a Canvas type exam. And in order to maintain as much security as we can, we're going to implement this lockdown browser this semester. I'm going to put instructions for you up on Canvas next week for how to download this thing and a practice quiz for you to try using the lockdown browser, so that we'll make sure that everybody is ready to use it before the first exam comes. So it's nothing to be worried about. It's just a tool that will help ensure that we have the best security we have while you're taking exams in the classroom this semester. I feel really, really strongly, my personal opinion is that I feel very strongly about protecting students who are honest. I will go to the end of the earth to protect students who are honest and who do honest work and take exams without any shenanigans going on. I want to protect them from students who cheat. I don't think it's fair if a student cheats and gets a higher grade than a person who works hard and does things honestly. Okay, so that's one thing to know about me. I'm a real stickler about that. I know a lot of you, this is an important course for you, particularly if you're heading into a nursing program or you're in a nursing program. So I want to make sure that you're not competing against somebody in class who is doing things unfairly. So keep an eye out next week and we'll talk a little bit more about Respondus. Just out of curiosity, do me a favor and put the number 1 in the chat if you have ever taken an exam using Lockdown Browser. Has anybody ever taken an exam using this application called Lockdown Browser? Okay, I didn't think so. This is an application that's in use at other colleges in New Hampshire. It's heavily used, but I'm pretty sure this is the first semester that we've used it at Keene State. All right, very good. All right, let me pull this up here. I'm going to share my screen again, but I'm gonna knock that down. Stay with me for just a minute. I'm just shifting things around here. All right, let me try again. Well, actually, before I do that, does anybody have any questions that I can answer? Does anybody have any questions about the syllabus, the course policies, what's expected of you this semester? Any questions about that? Anything in that Start Here module? Anything that's in our to-do list this week? Any questions? Okay, very good. All right, what I'm going to do now is pull up a slide set for our lecture that we're having today, which is just introduction to the microbes. I want to point out to you that for each one of our lectures this semester, I'm going to post the PowerPoint slides on Canvas for you. Now, the reason I do that is because some students like to pull those up on their own laptop during class. It's a lot easier for you as a student to take notes on the things that I say that are not on the slides. In other words, don't try to write down every single thing on every slide. You're going to drive yourself crazy if you try to do that. You have the slides. You have them on Canvas. So in your notes, you can write, slide number 1 and just write down anything extra that I say. Okay, any other additional information that I say about that slide. You don't have to write down what's on the slide because you've got the slide. You have the slide on Canvas. Okay, does that make sense to everybody? It's very hard to try to write down every single thing on every slide during our lectures. You're going to drive yourself crazy if you try to do that. So instead, in your notes, just write down slide number 1 and write down anything extra that I'm saying that's not on the slide. All right. And then when you get home and you're studying, you can just pull those slides up on Canvas. And as you go through them, you'll have your extra notes available and you can study. Okay. All right. I'm going to pull the slides up. So that's what you should be seeing on your screen right now, a title slide that says Introduction to the Microbes. One thing you should notice is that in the notes section, if you've ever used PowerPoint for your own presentations, there's a little section at the bottom of each slide called Notes. And sometimes I will put some information in the notes for you. Information that you might want to have in your notes. And this way you don't have to write it down because it's available to you in the slide. Okay. All right. So first things first, we're talking about microbiology this semester. So we need to understand what a microbe is because we're going to use that term a lot. The definition that we're going to use for a microbe is this. A microbe is a single-celled microscopic organism or acellular particle. A single-celled microscopic organism or an acellular particle. When we use the word organism, we're saying a living thing, something that's made out of a cell or cells. It's alive if it's an organism. An acellular particle is something that is not made of cells and therefore is not technically alive. Viruses are particles. They are not living things. They are particles. This sometimes freaks people out when you tell them this. Viruses cause terrible diseases in us, as we all know, having lived through the pandemic, but they are not cells. And on our planet, the only things that are alive are cells. Cells. So a microbe can be cellular, like bacteria or fungus, but it can also be acellular, without cells, a viral particle. When you think about all of the living things on our planet, all of them all together, as you may have learned back in elementary school, you can classify them. You know, you can put them in kingdoms and orders and phyla and families and so on and so on. The largest of those organizational tiers is called domains. So all living things on our planet can be broken down into one of 3 domains. And I've got them listed here: the bacteria, the archaea, and the eukarya. So it doesn't matter if it's an ant or a bacterial cell or a palm tree or a human being. Any living thing on this planet belongs in one of these 3 domains. Now remember, I also said there are things that we're going to talk about that aren't technically alive. The particles. The particles include viruses and another disease-causing particle called prions. So living things in the domains. Non-living things are the particles. You'll notice I have an asterisk next to bacteria and viruses. Those are the 2 organisms that we're going to, or 2 organisms and particles that we're going to talk about the most this semester. We're going to talk the most about bacteria and about viruses. And that's because in practical language, those are the 2 things that we deal with the most in microbiology. So the 3 domains of life, bacteria, archaea, eukarya, are not the only way that we classify things. As I said earlier, you probably learned in elementary school that we have kingdoms and we have phyla and families. We even have genus and species for every organism. But we talk a lot about these domains in microbiology because they help us keep things straight in our minds about the different kinds of cells that each of these different organisms has. So the type of cell that a bacteria is, the type of cell that an archaea is very different from the type of cell that a eukaryote is. Now, you and I belong in the eukarya domain. We belong with the protists and the fungi and the plants and the other animals. On this slide, you're looking at a drawing of what looks like a tree. Sometimes we call these drawings the tree of life. Technically, they are called phylogenetic trees. I'm going to type that word right here for you. Phylogenetic trees. These drawings that look like trees, these are what are evolutionary histories of living things. And if you've never looked at one of these phylogenetic trees before, you have to read them a certain way. You start right here at the base of the tree. What this represents to us is the first living thing that appeared on planet Earth. We sometimes call it the universal common ancestor, because there was one point in the Earth's history when the first cell evolved. Early in our history, there was no life on this planet. It was a very hot place. It was very volcanic. There were lots of toxic gases, and no, it didn't support life. But eventually, about 4 billion years ago, the conditions on the planet started to change, and the very first cellular life forms evolved. So we refer to that, whatever it was, that original cellular life form, as our common ancestor, because it was from that cell that all other life came about across time. So if we think about our domains on this tree, we're going to follow up from the base of the tree to the first branching point right here. That original cellular life form divided at some point. It split, it separated into 2 groups. One group evolved this way and became the bacteria. The other group went in a different direction evolutionarily, and eventually got to this point where it split again. Half of it, well, I say half, but some of it evolved into what are called archaea now, and the rest evolved into the eukaryotes. So from an evolutionary standpoint, the bacteria are the oldest domain on our planet. They evolved first. If you look down in the notes section, you can see some actual numbers. We know that life first emerged, as I said, about 3.5 billion years ago to 4 billion years ago, somewhere in there. That was that first common ancestral life form. And then about 3.5 billion years ago, the bacteria evolved. About 2.5 billion years ago, the archaea evolved. And about 2 billion years ago, the eukarya evolved. So bacteria came first. And then later on, about 1 billion years later, the archaea appeared. They emerged out of some event that occurred on the planet that caused some of those bacteria to keep evolving. into something different. And that different thing was an archaea. And then after another 500 million years or so, some event occurred that caused the eukarya to evolve, to emerge or appear on the planet. So we are actually in the youngest domain on planet Earth. All of the eukaryotes, the plants, the animals, the fungi, and the protists, we are the youngest domain, and bacteria have been here the longest. Now that tells you a little something about bacteria. Since they've been here the longest, on this planet the longest, that tells you that they have adapted very well on our planet. They've been very successful in terms of evolution. They have structures and functions that make them very adaptable to life on this planet. Think of all the changes that have occurred through our history. The bacteria came through all of them. They survived all of them. They didn't go extinct. The archaea came next. The archaea are just a little different from the bacteria. And then the eukarya came. They're even more different. But those three domains are the domains that we can put any living thing into that lives on our planet anyway. Now, I've said a couple times that cells are alive. There's something in, sorry about that. I have two dogs that are that love to pretend to be police officers and police the neighborhood. There's something in science that we call the cell theory. And it also emerged from evolutionary data, just like the domains and that tree of life. The cell theory tells us two really important things about life on Earth. It tells us that all living organisms are made out of cells. It also tells us that all cells come from other cells. All cells come from pre-existing cells. In other words, cells don't just appear out of nowhere. A cell has to come from another cell dividing. So remember, we talked about that first life form on the planet being a cell. That cell would have had to divide to make more cells. And then those cells divided and those cells divided and so on and so on and so on until today, when our planet is covered in cellular life forms, including you and I. And we are multicellular. We're made out of trillions of cells. But every single cell came from another cell. And that's an important thing to know, because that means that every new cell inherits things from the cell that created it. Every new cell that's produced inherits some genetic information from the parent cell that divided and created it. So the cell theory is an important thing for us to remember as we talk about microbes. All living things are made of cells, and all cells come from another cell, a previously existing cell. They can't just be created out of thin air. When we look at cells, when we look at the cells that are in the three domains, the bacteria, the archaea, the eukarya, they all are a little bit different. They all have different structures and different functions. But having said that, they also share certain characteristics. There are four 4 characteristics that every single cell has. It doesn't matter if it's a bacterial cell or an archaeal cell or a plant or an animal or a tree or a tiger. It doesn't matter. All cells have 4 things in common. Number 1, all cells are surrounded by a membrane. Now, cell membranes go by different names. Sometimes they're called cell membranes. Sometimes they're called plasma membranes. Those are referring to the same structure. It's a membrane that surrounds the cell, and it's made out of primarily 2 kinds of molecules. One is called a phospholipid, and the other one is called protein. Every single cell has a membrane, and every single cell has a membrane full of phospholipid and protein. What makes one cell different from another with regard to the membrane is what types of phospholipids it has, what types of proteins it has. So for example, in your body, the cells in your skin have a different membrane than the cells in your retina or the cells in your liver. They all have a membrane, but the membrane is a little bit different from one cell to another. It has different phospholipids and it has different proteins in it. So all cells have a membrane. That's number 1. All cells also contain this sort of gel-like material called cytoplasm. That term cyto, anytime you see that in a word, that just means cell. So cytoplasm refers to this watery sort of gel-like material that sits inside of every cell. All cells use DNA as their genetic material. And finally, all cells contain a molecule called ribosomes. Ribosomes are the machinery, if you will, that help build protein. We're going to talk about the process of how cells make protein later on in the semester. But understand that every single cell contains this molecule called a ribosome. They have many ribosomes. And these ribosomes are just little machines that help build proteins. Now, there's a couple of things we can point to in that list to support what I told you earlier about viral particles, right? Cells are alive. Viral particles are not alive. Cells have a membrane. Viral particles don't have a membrane. As we'll talk about later in the semester, viral particles are made out of genetic material and protein. That's it. That's all they have. They're a piece of genetic material surrounded by a coat of protein. That's it. So no membrane, no cytoplasm, no ribosomes. They do have genetic material, but only some of them have DNA as their genetic material, right? The SARS-CoV-2 virus, the virus that causes COVID, it doesn't have DNA, does it? That virus uses RNA as its genetic material. It uses a sister molecule to DNA called RNA. So it doesn't meet our definition for a cell. It just doesn't have the things in it, viral particles. They don't have the things in it that cells have. They don't have their own membrane. They don't have any cytoplasm. They don't necessarily use DNA. And they don't have any ribosomes. So people say, well, how in the world can those particles persist? How can they exist? And the answer is They hijack a cell. They get into a cell and they use that cell to replicate. They don't have what it takes to copy themselves, to reproduce themselves. So they come inside of a living cell and they let that cell make copies. If you think about it, it's a pretty good system. It's a sneaky little system. They're parasites. They get into our cells and our cells copy them. Our cells reproduce them. On this slide, you're looking at, on the left-hand side, a drawing, and on the right-hand side, an actual image taken with a microscope. So over here on the left, this cell down at the bottom represents a eukaryotic cell. So this would be a cell that comes out of the eukarya domain. It gets a little confusing in the language, but the domain is called eukarya. The organism is called a eukaryote. The thing that distinguishes eukarya from the other domains is that eukaryotic cells have these little internal compartments that you can see in here. These are called organelles. For example, there's a nucleus in here. There are mitochondria in pink. There's what's called endoplasmic reticulum. There's Golgi apparatus. There's all kinds of little compartments in here called organelles. Now, up at the top, you're looking at a different kind of cell. This is a cell that might have come out of the bacteria domain or the archaea domain, because it turns out that both bacteria and archaea are prokaryotic organisms. Prokaryotes don't have any organelles. They don't have a nucleus. They don't have mitochondria. They don't have endoplasmic reticulum. So we have prokaryotes and we have eukaryotes. Bacteria and archaea are prokaryotes. The eukaryotic organisms are all in the eukarya domain. Does that make sense to everybody? That's a really important point. There are 3 domains of living things: bacteria, archaea, eukarya. The bacteria and the archaea are called prokaryotes. We call them that because the type of cells that they have don't have any internal membrane compartments called organelles. Eukarya, that domain, contains eukaryotes. That's the kind of organisms. And those cells do have organelles. So prokaryotes and eukaryotes are different from each other because prokaryotes don't have organelles and eukaryotes do. Those cells are going to function differently. Those cells are going to work differently because of the fact that one type doesn't have organelles and one type does. If you look on the right-hand side of this slide, you can see actual microscopic images of these 2 general types of cells: eukaryotic cell on the bottom and 2 prokaryotic cells up at the top. These could be bacteria or archaea, we don't know. This one is just something that is in the eukarya domain. Notice that you can see the inside of this cell, and you can see it in a lot of detail, can't you? This image was taken with what's called an electron microscope. Electron microscopes can magnify cells tremendously, much more than regular microscopes can. You can see the nucleus in the center. And then you can see all of these little membrane compartments. They have a little bit of membrane around the outside, and there's a little compartment on the inside. These are all the different organelles. You can also see the cell membrane or the plasma membrane around the outside. Now, compare that to these two cells. These are prokaryotic cells. There are no organelles in here. Now, you can still see stuff in here. You can definitely see stuff. Let's think about what all cells have. All cells have a membrane. So there's a membrane around each one of these cells. All cells have cytoplasm. So there's cytoplasm in here. All cells have DNA. Now, in this cell, the DNA is in the nucleus. But in these two cells, there is no nucleus because they're prokaryotes. Instead, the DNA is sort of loose in the cell. So where you're seeing this sort of gray material in here, that's genetic material, that's DNA. And down here, you can see it as well, sort of grayish black looking material in that cell floating around in the cytoplasm. Remember, the fourth thing that all cells have are ribosomes. And there are ribosomes in all of these cells. They're just hard to see because they're very small. They're individual molecules, remember. So they're actually visible as little tiny, tiny black dots in the cytoplasm of all three of these cells. We find prokaryotic cells and prokaryotic organisms in two domains, the bacteria and the archaea. Prokaryotes are single-celled, microscopic organisms, and they have no organelles. That's how we define a prokaryote. It's a single-celled organism, one cell only. Single-celled organism, microscopic, so you cannot see it with your naked eye, and it has no organelles in it. That's what a prokaryote is. And you're going to find prokaryotes in the bacteria domain and in the archaea domain. Single-celled, microscopic, no organelles. All cells, we said, have DNA as their genetic material, and prokaryotes have DNA. Interestingly, their DNA looks a lot different from our DNA. It's the same molecule, but in a prokaryotic organism, the DNA is found in a single circular chromosome. You and I have what we call linear chromosomes. You and I have our DNA in a whole group of linear chromosomes, essentially lines of DNA, strands of DNA. Does anybody know how many linear chromosomes are in human cells? If I went into your body and I took a cell out and I took the nucleus out and I took the DNA out, how many chromosomes would I find in that cell? Type it into chat if you think you know. How many chromosomes are in human cells? How many linear chromosomes? Anybody have any idea? Good, good job. Yep, Sarah's got it. Jade has got it. Jenna, Rebecca, Caitlin, Josie, good. We actually have 46 chromosomes, but we often talk about them as being 23 pair of chromosomes. So we have 23 different types of chromosomes, but we get 2 copies of each one. You get one copy from your mom and one from your dad. So you have 46 total, 23 pairs. Good job. Now, the DNA inside a prokaryotic cell, it's floating around in the cytoplasm, but it's not floating around all by itself. Listen, DNA is a very stable molecule, but in a living cell, DNA spends most of its time wrapped around little balls of protein. These proteins in prokaryotes are called histone-like proteins. They are just proteins. They're just balls of protein that the DNA molecule is wrapped around. And then all of those balls of protein and DNA sort of compact together to form a structure that's called a nucleoid. They don't have a nucleus. They have a nucleoid, a big ball of DNA and protein. Now, here's something that I find very interesting about prokaryotes. In addition to their one chromosome, their one circular piece of DNA, they also sometimes have little small pieces of DNA called plasmids. Now, plasmids are called extra chromosomal DNA. In other words, outside of the chromosome. That's what extra chromosomal means. They're not part of the DNA that is necessary for the cell to survive. They are literally extra pieces of DNA. Now, not all prokaryotic organisms have plasmids, but a lot of them do. And what makes plasmids really interesting is that while they're not necessary for survival, when a cell has plasmids, those plasmids often give the cell very special properties. For example, when bacteria are resistant to antibiotics, that resistance comes from a gene that's found on a plasmid, a little extra piece of DNA, and it's usually a circular piece of DNA that bacterial cell happens to have. Not all bacteria are resistant to antibiotics, right? Thank goodness. But there are some bacteria that are resistant to our antibiotics. And the reason they're resistant is because they have this little plasmid. And the plasmid, because it's made out of DNA, has a gene in it. And that gene makes them resistant to antibiotics. So plasmids are kind of cool. The scary thing about plasmids is that bacteria can share them with each other. Now, you and I, we share our DNA, right? We share our DNA with our offspring. When we reproduce, we give a copy of our DNA to our offspring, to our children. That's what we call vertical transmission from parent to child. But what bacteria can do, not only can they give a copy of their DNA to their children, to their offspring, but they can also share their plasmids with neighboring cells. They can share their plasmid DNA with other bacteria that just happen to be in the area. It's pretty scary because that's how antibiotic resistance can spread sometimes within a bacterial population. We're going to spend a little time later in the semester talking about exactly how bacteria do that. But it's pretty neat. It's a neat trick if you think about it, to be able to share a little bit of your DNA with a neighbor, not a family member, with a neighbor. Now, when you look at that single circular chromosome inside of a prokaryotic cell, the truth is it's a lot simpler than our DNA. We have linear chromosomes. We have 23 pair of linear chromosomes. We have a huge DNA molecule. If you compare our DNA molecule to a prokaryotic cell, to a bacteria or an archaea, theirs is much smaller and much simpler. Now, what do I mean by that? Does anybody have any idea how big human DNA is? If you think about DNA, remember it's a double-stranded molecule. We call it a double helix, and it has bases in it. And on each strand, those bases sort of stick off and they bind to each other. They pair with each other. We call them base pairs. A, or adenine, binds with T, or thymine, and they make a base pair. Does anybody know how many base pairs are in human DNA? Do you have any idea how big our DNA molecule is? It's enormous. Any ideas? There are 3 billion base pairs in your DNA. That's obviously not an exact number, approximately 3 billion with a B base pairs in your DNA. You are a very complicated organism and you have a huge DNA molecule because that's what it takes to build and maintain a complicated organism like a human. Now, prokaryotes, they're not complicated. They're just one little cell, right? So their DNA is much smaller. For example, in the bacterium called Escherichia coli, or what we usually refer to as E. coli, you've probably heard of E. coli, there are 4.6 million base pairs. 4.6 million compared to 3 billion in human DNA. They're just a simpler organism, so they have a smaller piece of DNA in one chromosome. Now, let's switch gears and think about eukaryotic organisms. The eukaryotic organisms are found in one domain only, and that's the eukarya. Eukaryotes can be single-celled, or they can be multiple-celled or multi-celled. Eukaryotes can be microscopic or macroscopic. So much different from the prokaryotes. The prokaryotes are always single-celled, microscopic, no organelles. Eukaryotes can be single-celled, microscopic, but sometimes they're multiple-celled, microscopic. Sometimes they're multiple-celled, macroscopic. And all macroscopic means is visible with the naked eye. You and I are multi-celled, macroscopic organisms. We are visible without a microscope. So a much wider variety of organisms in eukarya than in prokarya. Prokaryotes don't have any organelles. Eukaryotes do. And all an organelle is a little membrane-bound compartment inside the cell, including things like the nucleus. Eukaryotes have multiple linear chromosomes. Remember, the prokaryotes have one chromosome, and it's in a circle. We have many, eukaryotes have many, and they're in lines or strands. Now, interestingly, the DNA in our chromosomes is also wrapped up in protein, but it's wrapped up in histone protein. Remember, we said in a prokaryotic cell, the DNA is wrapped around histone-like proteins. We have true histone proteins interacting with our DNA. When you put DNA and histone protein together in a eukaryotic cell, you create a molecule called chromatin. Your DNA, most of the time, exists as chromatin inside of your cells. Now, remember we said there are eukaryotic organisms that are single-celled and microscopic. Think, for example, about all the little things that live in pond water, or in a lake, or in a stream, or in the soil. There are single-celled microscopic eukaryotes. And some of them, not all of them, but some of them have those little plasmids, those little extra pieces of DNA in them. They're quite rare in eukaryotes, but you can find them periodically in some single-celled eukaryotic organisms. Multi-celled eukaryotes don't have plasmids. You and I don't have any plasmids. As we said, our genome, our DNA molecule is much larger and much more complex, many more base pairs, many more genes. We said that humans have 3 billion base pairs in their DNA. Does anybody have a sense how many genes you have in your DNA? Do you have any sense for how many genes human beings have? I'll give you a hint. We don't know the exact number yet. We're still working on that in the laboratory. The current estimate for how many genes are in human DNA is about 30,000. 30,000 genes in our DNA. We have only found a fraction of those. We've only investigated and studied a fraction of those genes. So we have about 30,000. A little bacterial cell probably has about 4,000 or 5,000 genes. So quite a difference there too. We're just more complex. So prokaryotes, single-celled, microscopic, no organelles. Let's take a minute and think about bacteria and how they're a little bit different from other prokaryotes. When you look at a bacterial cell, and we're going to have a chance to do this during the course, that cell membrane that all cells have is actually surrounded by another layer. In fact, some bacteria have multiple layers of material outside of their cell membrane. Bacteria have, outside of the membrane, a structure called a cell wall. And in bacteria, that structure is made out of a special kind of carbohydrate called peptidoglycan. Peptidoglycan. It's a carbohydrate. It's a polysaccharide. That's a technical term for a type of carbohydrate. And it's an extremely strong molecule. When bacterial cells divide, because that's how they make new bacteria, they use a process called binary fission. That's different from what you and I do, because of course we reproduce sexually. We take male and female together to reproduce, but bacteria just divide using this process called binary fission. This is a form of asexual reproduction. One of the interesting things about bacteria is that they use very diverse sources of energy. Now, by energy, think of food. They use very diverse sources of food. There are some bacteria that use organic molecules for food. There are some that use inorganic molecules, and there are some bacteria that use sunlight. There are some bacteria that are photosynthetic, just like plants are. Now, what do I mean when I call something an organic molecule versus an inorganic molecule? I'm not talking about the organic term that we use for our foods. When I use that word organic, what I'm talking about is carbon containing. In science, a molecule is considered an organic molecule if it has carbon in it. And of course, life on planet Earth is carbon-based. We're all made out of carbon and other types of elements. But there are inorganic substances on the planet. There are substances that don't have any carbon in them. Think about rock, for example, or sand. There's no carbon in there. There are some bacteria that can use inorganic molecules as food. All right, that's quite interesting because you and I can't do that. We need organic molecules as our food. And there are some bacteria that use sunlight. There are photosynthetic bacteria. They act like green plants act, and they harvest energy from the sun. So they're quite diverse in their ability to make use of different food sources. Now, when you compare the bacteria and the archaea, we see some important differences. The archaea are also prokaryotes. They are single-celled, microscopic. They don't have any organelles. But outside of their cell membrane, instead of finding that molecule called peptidoglycan in the cell wall, we find another structure called an S layer. So it's a layer outside of the cell membrane, but it's not made out of peptidoglycan. It's made out of protein and a type of molecule called a glycoprotein. Now, anytime you see this term glyco in a word, it means sugar or carbohydrate. So a glycoprotein is a protein that has a little bit of carbohydrate attached to it. So archaea don't have a cell wall. They don't have a peptidoglycan cell wall. Instead, they have a layer called an S layer outside of their cell membrane. They also use binary fission, that asexual process to divide, but archaea can do other things as well. There are some archaea that use a reproductive process called fragmentation. And some archaea use a process called budding. And there are other reproductive mechanisms that certain archaea can use. So they are more diverse in terms of how they reproduce compared to bacteria. Just like the bacteria, they can use organic molecules. Some of them can use inorganic molecules, and some of them can use sunlight for their food sources. What makes the archaea very interesting scientifically is this last point down here. The archaea have evolved to be able to live in some of the most extreme environments that you find on our planet. Unlike the bacteria, archaea can live in acid. They can live in high salt concentrations. They can live at very high temperatures. They can live at very low temperatures. Now, not all archaea can live in all environments. There are individual species that can survive in acid. There are individual species that can survive in high salt. There are species that can live at very high temperatures. There are some that can live at very low temperatures. So remember, we said the bacteria evolved first on planet Earth, and out of the bacteria came the archaea. And what the archaea did was adapted themselves to some of the most extreme environments on our planet, places where no other living thing can survive. It's really fascinating. The other thing to know about the archaea, and this is an important one, put a star next to this in your notes. While there are some bacteria that can cause terrible disease in humans, there are no archaea that cause disease in us. Isn't that interesting? There are no pathogens that are archaea. I think that's really interesting. One of the words that you'll sometimes hear applied to archaea is this one over here. They're called extremophiles. Now, if you break this word down, if you translate it, the extremo refers to these extreme environments that some of them can live in. And the phile means loving. So this word translates as extreme loving. They love extreme environments. Now, it's important to note that there are some archaea that don't live in extreme environments. There are some archaea that live in perfectly ordinary places like ponds and streams and the soil. In fact, there are archaea that live on and in your body. There are archaea that are part of your normal microbiome. They're perfectly normal to have on you and in you. They don't harm you at all. And they may actually help keep you healthy. So the archaea are nothing for us to be afraid of or nervous about because they just don't cause disease in us. I have some more of those high-powered microscopic images on this slide. These images were taken with an electron microscope, and I put them in here to show us the difference between a cell wall and an S layer. Now, you can't see a lot of detail on the top row of images. You can see the cells though, and you can see their shapes. Bacteria and the other prokaryotes have a limited number of shapes that they take in their cells. For example, in this image here on the left, you can see two cells here, two bacterial cells side by side. And these cells are spherical, aren't they? They're round. They look like little balls. We call this shape a coccus, and we would say these are cocci. These cells in the middle, these are rod-shaped bacterial cells. And then over here on the right, you can see a very strange looking bacterial cell. This is called a spirochete. It literally looks like a spiral. So if I were to touch these cells, if I were capable of touching these cells, what I would be touching is the cell wall. The outer layer, what's outside of the cell membrane in bacteria, is called a cell wall, and it's made out of peptidoglycan. Now, down here on the bottom, I have an even higher magnification of an archaeal cell. So you can see this one is also rod shaped. I just have a higher magnification of it. This is the S layer. I want you to look at the detail on it. Here's some more S layer over here. It almost doesn't look real. When I look at it, it looks like this little cell is wrapped in an ace bandage or something. It's got all kinds of texture on it. The S layer is outside of the cell membrane in an archaeal cell. Cell wall in bacteria, S layer in archaea. On this slide, we're just looking at some of these extreme environments that I mentioned. This up here, this is an actual place. This is an actual structure in an actual place. You can find this in Yellowstone National Park. It's a pool of water. It's called the Morning Glory Pool. And the reason it has this beautiful blue color to the water is because it has a lot of different organisms living in it. Same thing with this golden color around the edges. There are all kinds of prokaryotic cells growing here. I think you can just see there's some steam coming up from this water as well. This is like a hot spring. Now, over here, what you're looking at are some vents on the floor of the ocean. So very deep down in the ocean, you'll find these strange structures called thermal vents. And they are venting out hot gases from deep down in the Earth's crust. There are archaea that can live inside of these vents. There are no bacteria that could survive that kind of heat, but there are some archaea that can. And over here, this is another place. This is actually found in Spain. It's a river that very sadly got terribly contaminated with heavy metal. And over time, the water turned this brownish color and it became extremely acidic because it's full of heavy metals. So no human could touch this water without getting injured. But there are archaea that live in this little river. There are archaea that live in that high acid, heavy metal contaminated soup. Now, let me stop here for a minute and ask if anybody has any questions so far. Any questions about what we've talked about in terms of prokaryotic cells, eukaryotic cells, what the differences are, the things that cells share in common, and so on. Remember, we said at the very beginning of the lecture, a microbe is a single-celled microscopic organism or particle. So right now we can say that prokaryotes are microbes. Some eukaryotes are microbes, right? There are some eukaryotic organisms that are single-celled and microscopic. Think about things like amoeba and other types of protozoa that you might find out in the environment. There are single-celled microscopic eukaryotes, and those would be called microbes. And of course, viruses are also called microbes, even though they're not cellular, they're acellular. I do want to say just a couple things about the fungi. Remember, in the eukarya domain, we have plants, animals, protists, and fungi. Fungi, like other eukaryotes, are sometimes single-celled and sometimes multiple-celled or multi-celled. Sometimes they're microscopic and sometimes they're macroscopic. What's interesting about the fungal type of cell is that it also has a cell wall, but its cell wall is made out of a different kind of carbohydrate than bacteria or archaea. It's made out of a carbohydrate called chitin. Now, you can find chitin in other organisms as well. You can find chitin, for example, in insects. Any insect that has a hard shell around it, an exoskeleton, that hard shell is made out of chitin. It's just a carbohydrate. It's another one of these polysaccharides, but it's something that you don't find in humans. We don't have any chitin in our bodies. Fungi can use both asexual reproduction and sexual reproduction. It depends. It depends on the species, and it depends on what point in their life cycle that they're at. So they are very complex organisms. Unlike the prokaryotes, fungi are limited in what kind of foods they can use for energy. They can only use organic molecules. You and I are the same. Animals, like humans, are also limited to organic molecules as food sources. And the fungi are like us that way. Remember, the bacteria and the archaea can use all kinds of things. They can use inorganic, and some of them can use sunlight. But the fungi are limited. They're limited to organic molecules. We don't talk a lot about fungi in this course. We spend most of our time talking about bacteria and viruses. But there is something you should know about the fungi, and that's down here at the bottom of the slide. There are 3 general categories of fungi that we see, that we encounter. The first ones are called yeasts. The second are called molds. And the last is called mushrooms. So obviously mushrooms are macroscopic. We can see them with the naked eye. They are also multicellular. Mushrooms are actually reproductive structures. So the fungus produces a mushroom in order to reproduce, because the mushroom contains little reproductive structures within it. Yeasts and molds are different. Yeasts are single-celled, microscopic fungi. So yeasts are microbes. Molds are different. Molds are multicellular, and they can be microscopic or macroscopic. We've all seen mold growing, right? We've all seen macroscopic mold, unfortunately. We've all seen macroscopic mold growing in the bathroom, right? Around the edge of the tub or wherever. Mold can grow to the point where it's visible with your naked eye. It can also be microscopic if it hasn't grown very much. In other words, you would need a microscope to see it. But mold is a multicellular type of fungus. Only yeast is considered a microbe because yeast is single-celled and microscopic. One of the things that we'll talk about when we talk about laboratory testing is how hard it is to tell a fungus from a bacterium in the laboratory. What you're looking at here on the upper left, this is a Petri dish that has some growth media in it. And you can see a whole variety of different fungal species growing on this plate, on this agar plate. Now, when we see something that has a greenish blue color growing, whether it's on an agar plate or on a loaf of bread, we often think about fungi because they just have a characteristic look to them. They often have a green-blue color. They're often sort of fuzzy looking. They have irregular borders to them. But the truth is, you cannot tell a fungus from a bacterium until you look at it under the microscope. Over here on the right-hand side, you're looking at one of those high-powered electron microscope images. These are yeast. These are yeast cells, single-celled, microscopic fungi. Notice that they're not perfectly round. They're kind of oval shaped. And I want you to notice that this cell right up here, this big cell, has a little tiny cell attached to it. This big cell is reproducing, and it's reproducing in a mechanism called budding. Fungal yeast will produce new cells by budding them off of their surface. And this little baby yeast cell will grow up to be its own unique independent cell. Down here, this is just a box full of different types of mushroom species. Gosh, you can find so many different mushrooms growing in our state. And of course, it's so important that you never eat wild mushrooms. It's very, very hard to know which mushrooms are safe to eat and which ones are toxic. And toxic mushrooms will kill you within about 24 hours. So I always tell people, don't ever, ever pick mushrooms in the forest unless you're with an expert. Go to the grocery store for your mushrooms. Don't run that risk. Don't risk eating a toxic mushroom. The other type of microbe that we sometimes see in the eukarya domain are what we call protists. Now, the first kind of protist I want you to be familiar with is called a protozoa. Protozoa. If you translate this word, it means primitive animal or first animal. Protozoa. Now remember, what makes an animal is its ability to move. So these are microbes, these protozoa, because they're single-celled and they're microscopic, but they can move. They are either free-living or parasitic. We do see some parasitic protozoa. And they have multiple life stages. They go through multiple life stages before they're adults. And they're mobile. And that tells you that they have to have a means of movement in order to allow them to move through their environment. They're eukaryotes. They don't have any kind of cell wall, though, just a membrane around them. So unlike the fungi that have chitin in their cell wall, protozoa don't have cell walls. They can use both asexual and sexual reproduction methods. And like the fungi, they can only use organic food sources. So amoeba would fall into this group. You may have heard of a parasite called Giardia. That's in this group. There's one called Toxoplasma, also a pathogen, and one called Trichomonas that also causes disease in humans. If you look at the images over here on the right, this is an amoeba. Everybody learns about amoeba in elementary school. Amoeba are able to move because they're able to send out extensions from their membrane. They literally push out their membrane into these little sort of feet, and they use them to crawl through their environment or swim through their environment. This right here, this is Giardia under a microscope. Giardia, to my eyes, looks like it has a pair of eyes and a nose. The weirdest thing about Giardia is that it has multiple flagella. It has multiple little tails, these long hair-like structures that are coming off of it. These are flagella, and that helps Giardia swim through its environment. Giardia is usually contracted from freshwater sources. So people go camping in New Hampshire, and every year there are cases of Giardia because sometimes people look at a stream or a river and they think, oh, it must be perfectly pure water because we're in the woods and it looks pretty, and they'll scoop water out of the stream and they'll drink it. And Giardia lives in fresh water. You don't want to get Giardia. If you've ever known anyone who's had Giardia, it causes a terrible intestinal infection, terrible diarrhea. You can take medicine and recover from it, but it's not the kind of infection you would want to get. So you never want to drink water out of a wild source without purifying it somehow, preferably by boiling it. This little critter right here that you can see on this image, this is Toxoplasma. This is a protozoa that can cause disease, particularly problematic in pregnant women. You've probably heard of how pregnant women shouldn't be changing their cats' litter boxes because cats can get this toxoplasmosis disease as well, and they will shed these organisms in their feces. So pregnant women are often advised not to change the cat litter box. You'll also find toxoplasma out in the soil. So it's very important to wash your hands after you've been digging in the soil. And this little guy down here, who almost looks like a giardia, looks a little bit like a giardia. This is a protozoa called Trichomonas. And this little organism causes a sexually transmitted disease. So protozoa, these are primitive animal life forms. They're capable of movement, and we consider them microbes because they're single-celled and microscopic. The other ones that you should be familiar with are the algae. Algae are protists. Algae, like fungi, like the fungus, they can be single-celled or multi-celled. They can be microscopic or macroscopic. They also have a cell wall. But what's weird about the algae is there are so many different types of algae. They all have a different type of material in their cell wall. There are cell walls made out of a molecule called carrageenan. There are some made out of a molecule called agar. There are all different kinds of cell walls around algae cells. Algae can use asexual or sexual reproduction, depending on the species. And algae are photosynthetic. We used to put algae in the plant category because they're photosynthetic. They get their energy the same way that green plants do. But we now know that algae are very different from plants in other ways. So while they are photosynthetic, they're considered protists instead of plants because they're just very different from the green plants. On this slide, I just wanted you to see some drawings of all the different types of algae. There are microscopic algae, there are macroscopic algae, there are single-celled algae, there are multi-celled algae. There are many, many different types, different species. There are what we call green algae, there are red algae, and there are brown algae. Lots and lots of varieties. And of course, there are also acellular microbes. We are going to talk about viruses and a little bit about those prion particles, but we're not going to talk about them today. We're going to save that for another day. All right. So we're out of time today. So that's where we're going to finish it up. When we get together on Thursday morning, we're going to do our lab exercise. So when you go onto Canvas and you go into the week 1 module, you're going to find the materials that we're going to use in lab on Thursday when we get together over Zoom. You're going to find the set of slides that I'm going to be using. And you're also going to find a handout about the care and use of the microscope. Everybody comes to this class with different experiences handling microscopes. So we're going to go through the different parts of the microscope just to make sure everybody's on the same level of understanding. Does anybody have any questions that they'd like me to answer before we break for the day? If not, I want to thank everybody for coming. And I will see you again on Thursday morning at 10 o'clock. If you have any questions, feel free to message me through Canvas. Okay, any questions, any problems, anytime you need anything from me, just send me a message through Canvas. All right. All right, everybody. I'm going to let you go. Enjoy the rest of the day, and I'll see you on Thursday.
Thank you.
Thank you.
Thank
You're
you.
welcome.
Thank you.
Thank you.